Hello, my name is Claudia Collier, and I'm a senior at the University of Texas at San Antonio. And today I will be presenting my project, Wireless Non-Invasive Force-Inducing Neuronal Stimulation Mediated by Magnetic Nanodiscs. So with diseases like Parkinson's, cell-type specific manipulation of neural circuits is required for treatment. There are existing therapies that are in place and in practice. For example, pharmacological agents. There is a manipulation of neural signaling that is restricted by the blood-brain barrier, the rapid cerebral fluid clearance, and there's also a lack of cell-type specificity, which creates side effects and treatment resistance. Then there's electrical stimulation, which may provide a way of repairing neural dysfunction. However, these solutions are invasive, non-specific to cell types, and there's a high possibility of surgical complications. So our area of research is looking at alternative methods, such as magnetic nanodiscs or MNDs. And these MNDs can be used for heat dissipation under alternating magnetic fields to control cellular signaling wirelessly. So our goal of this project is to develop a wireless, non-invasive, and cell-type specific platform that allows for the modulation of neuronal activity by transducing AMFs into mechanical stimuli. And we will employ magnetic nanodiscs as they are good transducers of low frequency, low amplitude AMFs into mechanical forces and can be used to evoke neural activity of motor cortical neurons. So in figure 1, here shows functionalized MNDs targeted the surface of the primary motor neurons expressing the GCAMP6. And upon exposure to an AMF, the torque produced by the MNDs triggers the response of the mechanosensitive ion channels. This response allows for an ion influx that is detectable through the fluorescence increase of GCAMP6. In order to carry out this experiment, MNDs composed of iron nickel permalloy and coated with gold were fabricated through the interference lithography techniques by our collaborators in Spain. In figure 2, you can see that our MNDs were surface functionalized with biocompatible oligoethylene glycol methyl acrylate, or known as POEGMA, through atom transfer radical polymerization. Functionalized MNDs were then layered on silicone wafers that underwent an extraction process, and then the cortical neurons were extracted from newborn rats and cultured with neurobasal media. Then after 1 week of culturing, primary neurons were transfected with the mechanosensitive ion channel of large conductance EMSCL using lipofectamine 2000 reagents. Then fluorophore calcium imaging was performed on primary neurons under AMS stimulation. So here, the characterization of MNDs in these figures, it shows the average size of the MNDs with POEGMA to have a thickness of 153 nanometers and a diameter of 922 nanometers. And in figure B, the DLS calculated the average charge of the MNDs with POEGMA to be negative 103.5 millivolts without the polymer and 18.8 millivolts with the polymer. In figure 4, we have confocal images with MNDs in fixed cortical neurons. So on the far left, we have a bright field image with the MNDs in black. Then stained in magenta is the beta-3 tubulin, the cytoskeleton microtubules. Then the synopsis channel is stained yellow with mouse monoclonal synaptophysin. And then on the far right are the channels merged with the nuclei stained blue with DAPI. Then in figure 4b, we have SEM images showing the MNDs in white in close proximity to the neurons labeled in red arrows. Relating to the main portion of the project, in figure 5, we have the neuron activity and the likelihood of activity of cortical neurons. We set the video and the field to turn on at 30 seconds and automatically turn off after 1 minute, as can be shown in the purple shade of the graphs. So in set A, these cortical neurons undergo an AMF with the MNDs in the neurons. In set B, these neurons undergo no AMF, but they do have MNDs. In set C, these neurons undergo an AMF with no MNDs. And in set D, these neurons undergo no AMF with no MND. So with this data, we can see that there is more activity with the set A neurons that had both an AMF and MNDs compared to the others which either had none, or the other, or neither. And taking a closer look at set A, the neurons typically turned or showed fluorescent changes when the AMF was turned on and continued to show interactions with the mechanosensitive ion channel, even after the field was turned off. So in conclusion, we developed a wireless strategy for neural modulation, transducing magnetic signals into mechanical stimuli. So this work establishes the foundation for transgene-free and non-invasive magnetomechanical neuromodulation for the treatment of neurological disorders. And future work would address the functionalization of MNDs for cell type specific recognition. And this work has been supported by the MARC program and the GREAT grant from the VPR seed funding program. Thank you for listening. Do you have any questions?